Hi everyone, this is the National Taiwan University iGEM team from Taipei, and this year we created SAC, a standardized AI-powered platform for carbon sequestering spider silk production. The name of our project is inspired by spider egg sacs, silk structures woven by spiders to protect their eggs, with municipal waste generation rising every year due to excessive material discharge, a need for alternative materials that are biodegradable, environmental friendly, and on par with industry standards also arises. This is where SAC comes in. Spider silk has been proven by extensive research to be a remarkable natural material known for its extraordinary properties. It has unmatched mechanical strength, 5 times tougher than Kevlar, and 3 times more flexible than nylon, making it one of the most durable materials found in nature. With 41,000 spider species producing 7 types of silk each, it is also highly versatile and suitable for various applications. However, spider silk production faces 2 main challenges. First, there is a lack of understanding about the relationship between silk protein sequences and their mechanical properties. This limits our ability to customize spider silk, reducing the range of possible applications. Second, scaling spider silk manufacturing efficiently remains difficult due to the absence of standardized protocols for production, purification, and post-processing. Our project, SAC, directly addresses these challenges to accelerate the real-world application of spider silk as a sustainable biomaterial. SAC is made up of 3 key components at the upstream design phase. We have Generative Personalized Spider Silk GPSS, a software tool for generating customized spider silk sequences. By leveraging advancements in AI, such as GPT models, we designed GPSS to create sequences with tailored mechanical properties, allowing us to generate spider silk for specific use cases. Moving to the midstream biomanufacturing phase, we focus on sustainable carbon sequestering production. Once the custom silk sequence is generated, we produce it sustainably using a bioreactor that utilizes industrial waste, such as molasses, wastewater, and crude glycerol as a carbon source. Our life cycle analysis LCA model confirms the platform's carbon sequestering capabilities, offering both environmental and economic benefits through carbon offsets. Finally, at the downstream application phase, we focus on post-processing, applications, and recycling. After production, the spider silk protein is processed for various applications using 3 methods: fiber spinning, gelation for hydrogols, and film casting. These materials can be recycled by breaking them down into reusable building blocks, or by feeding them back into the bioreactor as a carbon source. Additionally, wastewater from the bioreactor can be repurposed for energy production, irrigation, and other uses. Through SAC, we ensure the production of high-quality recombinant spider silk, creating a path for scalable, eco-friendly applications in both industrial and medical fields, while supporting a circular and sustainable economy. Now let's introduce one of the core components of our SAC project, Generative Personalized Spider Silk GPSS. GPSS is an AI model designed to generate customized spider silk protein sequences customized to your specific needs. Think of our interface like a restaurant menu. Simply adjust 4 key properties: toughness, elastic modulus, tensile strength, and strain at break using simple sliders, each ranging from 1 to 10. Once you hit generate, our system processes your input in just 5 seconds, delivering a protein sequence optimized to meet your requirements. What's more, the information provided includes a comprehensive breakdown of predicted properties, amino acid composition, molecular weight, and even the corresponding DNA sequence, giving you everything you need to proceed with your research all at your fingertips. And if you're curious about how the sequence looks in 3D, it's as easy as clicking Generate again to visualize the structure. We've designed it to be as straightforward as possible. On the technical side, GPSS leverages powerful technologies like Python, Flask, and OmegaFold to ensure precise and fast results. Whether you're generating sequences or visualizing complex 3D structures, GPSS streamlines the entire process, allowing researchers to focus on innovation rather than technical details. We believe that GPSS bridges the gap between advanced AI and practical usability, opening up new possibilities in biotechnology and beyond. By making this technology approachable, we're empowering researchers to explore new frontiers without the complexity traditionally involved. Next, let's move on to the wet lab phase. Our wet lab efforts focus on simulating a seamless experimental workflow that starts with AI sequence generation, ends with application-ready spider silk-based fibers, gels, and films. By first establishing a clear lab-scale platform, we aim to standardize the complex process of synthetic biomaterial production and accelerate its eventual scaling for larger yields. To obtain accurate and verified ground truth, data for training GPSS, our LLM-based sequence generation tool, we also designed and performed specific property testing on our post-processed spider silk materials. Our wet lab process follows the iGEM recommended design build test learn cycle, aligning each stage of SAC's spider silk production with this framework. This workflow begins with AI sequence generation construct design in the design phase, followed by cloning, validation, fermentation in the build phase. We then move into the test phase with post-processing mechanical properties, and finally we close the cycle with the feedback loop for AI optimization in the learn phase. Let's dive into our SAC project. To make things easier to explain, we will use the T9E9TS9S1 sequence as our demonstration throughout. We used GPSS, Generative Personalized Spider Silk, an AI-driven platform, to design spider silk from scratch. This allows us to adjust the silk's mechanical properties like toughness T, elastic modulus E, tensile strength TS, and strain at break S to meet specific industrial needs. For example, the T9E9TS9S1 sequence offers high toughness and tensile strength, making it ideal for protective gear like bulletproof vests. Other sequences, such as T9E9TS9S9, combine toughness and elasticity, making them suitable for aerospace applications like parachute cords. We also designed sequences like T7E7TS7S3 for medical sutures and T3E3TS3S3S7 for structural reinforcement. To produce these silk proteins, we used the PET28A T7 MEG-FP plasmid, which includes a T7 promoter for high-level expression, a LACO operator for IPTG-controlled induction, and MEG-FP to visually track protein production. After preparing the sequence in plasmid, we moved to the build phase. Spider silk genes, whether natural or anticipated by our GPSS platform, are highly repetitive, which makes them difficult to synthesize directly. To overcome this complexity, we used PCR extension to gradually build the sequences and assembled them using Gibson assembly. Once the plasmid was constructed, we transformed it into NEB-stable E. coli cells and verified the sequence through double digestion and colony PCR. After sequencing confirmation, we inserted the plasmid into BLRDE3 E. coli, a strain optimized for protein overexpression. We set up LB medium flasks with FLEXS, our real-time data collection hardware device, to monitor key parameters such as pH, temperature, and optical density OD. Using the MEG-FP fluorescence intensity for quantification, we found that at 25 dC and pH 7, the protein yield was highest. Protein production was further evaluated using SDS page to assess expression levels, and Western blotting with MEG-FP antibodies confirmed the presence of the spider silk proteins. For deeper insights, we observed the formation of T9E9TS9S1 spider silk micelles using fluorescence microscopy. After confirming the protein's accuracy, we immediately began the fermentation process using wastewater. Once fermentation in the bioreactor was complete and the pH became alkaline, indicating nutrient depletion, we harvested and lysed the cells. This released the spider silk proteins, which were then purified and treated with SDS. After centrifugation and washing with deionized water, we achieved a final yield of 1.7 gl, marking a significant milestone toward large-scale production. For testing, we focused on two main post-processing techniques. First, fiber spinning. We used the fibers produced from the T9E9TS9S1 sequence for fabrication. Tensile strength testing showed that these fibers exhibited high tensile strength and flexibility, making them ideal for high-performance applications like textiles or military uses. Second, film casting. The spider silk HFIP solution was cast into Teflon molds to create films. In tensile strength tests, the films displayed lower tensile strength than the fibers but maintained strong correlations in strain and toughness, preserving the core mechanical properties of the protein. We validated our AI model's predictions through tensile testing, providing ground truths for improving our GPSS spider silk sequence generation process. Beyond mechanical testing, we needed to understand the silk's microscopic structure. In XRD experiments, the T9E9TS9S1 spider silk protein displayed sharper diffraction peaks and a higher degree of crystallinity compared to other samples. This suggests a highly ordered and closely packed molecular arrangement, which helps explain its superior tensile strength and toughness. Finally, we've arrived at the learn phase, where we reflect on the insights gained from our experiments. Our experiments confirmed the functionality of the SAC system, from AI-designed sequences to real spider silk materials. However, we observed inconsistencies during property testing, likely due to human factors in purification and spinning. We also noted that the MEGFP fluorescent protein's hydrophilic nature might interfere with the beta sheet structures that give spider silk its strength. On the positive side, we found that using wastewater in bioreactors was highly effective for large-scale production, moving us closer to sustainable industrial bioprocessing. With our wet lab experiments successfully demonstrating the feasibility of producing high-performance spider silk using AI-designed sequences and sustainable fermentation processes, we recognize that science doesn't exist in a vacuum. To truly make an impact, we needed to integrate our work with societal needs and values. This realization brought us to the human practices aspect of our project. First of all, our innovations in hardware and software contribute greatly to the iGEM community. For non-technical consumers as well as for researchers accelerating biomaterials innovation, GPSS Generative Personalized Spider Silk is a useful tool that helps both the customized spider silk proteins. Another one is FLEXS Flexible Extension Sensor System prevents contamination concerns, automates data collecting, and reduces human work. Gives future iGEM teams, particularly in biomanufacturing bioreactor-like capacity for small-scale fermentation condition optimization. In connecting SAC with society, we combine stakeholder engagement, partnerships, and expert advisory to gain valuable insights that guide us in building a shared vision for a more sustainable future. We focused on engaging various stakeholders, including students, environmental organizations, agricultural communities, policymakers, industry professionals, and fellow iGEM teams. Through presentations, workshops, and interactive events, we shared our project's goals and actively gathered feedback. For instance, we collaborated with MIT professors to upgrade our software tool, GPSS, and partnered with Microchip Technology to develop our hardware, FLEXS. In the early stages of our process, we worked with Taiwanese farmers to source wastewater samples for resource acquisition. During the process development phase, we consulted with experts in spider silk production. Additionally, we collaborated with Taipei University of Technology to develop automated fabrication machinery for post-processing. For product development, we engaged with professionals in the textile industry to understand market demands for sustainable materials. We also visited recycled fiber companies that showed interest in incorporating our spider silk into their products. Moreover, we worked with MICEL Technologies carbon auditors, and together we calculated that our wastewater-based medium reduces carbon emissions by 30% compared to commercial mediums. This approach addresses both wastewater and carbon emission issues. To ensure our wastewater-based production system was both efficient and sustainable, we consulted with domestic and international professors specializing in wastewater treatment and microbial production. Their insights guided us in selecting appropriate wastewater types, optimizing microbial strains, and improving the fermentation process to maximize protein yield while minimizing environmental impact. Finally, in sustainability education, we organized educational workshops and lectures for students from middle school to university, inspiring the next generation of scientists and promoting public understanding of synthetic biology. Interestingly, we also explored entrepreneurship as part of our project. After engaging with various stakeholders, we realized the commercial potential of SAC. So we applied for NTU's entrepreneurship grants and have been in discussions with local and international accelerators and investors. We even had the opportunity to consult with the CEO of DBS Bank to explore business possibilities. Regarding Sustainable Development Goals (SDGs), SAC aligns with several key objectives: SDG 6, SDG 7, SDG 9, SDG 12, SDG 13, and SDG 15. In our future work, the Wet Lab team will continue collaborating on the development of GPSS and the wastewater platform, while also advancing toward industrial-grade applications and exploring additional downstream uses, such as developing functional spider silk. The Dry Lab team will focus on further enhancing GPSS. We plan to establish a high-throughput spider silk production pipeline and create a comprehensive dataset on silk properties. This will support the development of a pre-trained model to better understand and analyze the characteristics of spider silk. In human practices, we will continue centering our efforts around AI and the UN's SDGs. We aim to build dialogues, consultations, and partnerships with stakeholders at all stages of the production process, from upstream AI development to midstream wastewater processing and downstream applications of spider silk materials, such as textile and material companies. Our goal is to establish collaborations with sustainable enterprises and ensure the long-term development of the SAC project, even beyond iGEM, contributing to the biomanufacturing industries in Taiwan and globally with a new, sustainable vision.